Hi everyone, my name is Lorenzo and today we're going to look at the GPCR pathway involving phospholipase C, diacylglycerol, inositol trisphosphate, and protein kinase C. So if you are interested in that, let's start. So the G-protein couple receptor that we looked at in a previous video, it's a 7 transmembrane receptor that's mainly for hormones and neurotransmitters. In this case we have glutamate, that's a neurotransmitter that binds to this G-protein couple receptor. And as with all G-protein couple receptor, we have a G-alpha subunit, so a G-protein associated with the G-protein couple receptor, and the G protein is composed of a G alpha subunit, a G beta, and a G gamma. And so the G alpha subunit is what we are interested in because G alpha GDP is inactive. So G alpha GDP is inactive and G alpha GTP is active. So how do I remember that? Well, GTP has more phosphate, so it has more energy, and so G alpha GTP is more active. Okay, and so when we have no ligand bound to the receptor, G alpha GDP is present, and so we have no activation. When there is the ligand that binds to the receptor, we have G alpha dissociation, and when we have G alpha dissociation from G beta gamma, this can go to cyclic AM adenylate cyclase, yes, or it can go to phospholipase C. So what is phospholipase C? Anyway, adenylate cyclase, we looked at that in the previous video. And in this case, we have this enzyme that's called phospholipase C. The name suggests that it is a phospholipase, so a lipase. Phospholipase, what does it mean? Well, that we have phospholipids. This enzyme will cleave these phospholipids. Okay, so phospholipase C cleaves phospholipids. And where are phospholipids present? In the membrane. So phospholipase C is in the membrane. Here we have phospholipase C. So phospholipase C takes a special type of that's activated by G alpha. So G alpha GTP goes and activates phospholipase C. In particular here, I don't know if you see, but it's the GQ alpha. So we have different type of G alpha, and in this case it's the GQ alpha. And the GQ alpha activates the enzyme, and the enzyme takes in phosphatidylinositol 4,5-bisphosphate or PIP2. And this is a phospholipid, as I said, that has two fat molecules, and then we have a glycerol, if you remember, and then the glycerol has 3 OH, but 1 OH is not like a triglyceride where we have a third. So in the triglycerol we have a third fatty acid, and in this case we have a phosphate, POO, and then attached to the phosphate we have an R group. That can be, for example, choline, or in this case, inositol. Inositol is a sugar-like molecule, and it's a little bit like glucose. It's derived from glucose. And so what the phospholipase C does is that it cleaves, I'm pretty sure that it cleaves here. So we have here, we have the inositol, and inositol has phosphates also attached to it, and so it gives inositol 3-phosphate, and this part is inositol 3-phosphate. So we have inositol with trisphosphate, and then we have diacylglycerol. So this is diacylglycerol with the glycerol attached because we have two acyl fatty acids, one glycerol, and so we have DAG, diacylglycerol. The name makes sense. Well, you may not know that, but the names make some logical sense. And then, so IP3 is a small molecule that in the cell opens. So here we have another membrane. This is the endoplasmic reticulum in particular. And in the endoplasmic reticulum, we have calcium channels, Ga channels. And as you may know, inside the endoplasmic reticulum, so here, we have more calcium. While in the cytoplasm, we have less calcium. So the gradient is in this sense. And the gradient is in this sense, so if we open calcium channels with inositol trisphosphate, calcium flows out. And if calcium flows out, it activates proteins such as calmodulin. So calmodulin, for example, gets activated. But there are many other proteins that are dependent on calcium. And so calmodulin can activate other proteins, and so we have a cascade that depends on the type of cell and hormone that we're talking about. So for example, here glutamate is an excitatory neurotransmitter inside neurons, so it may increase, for example, neuron excitability. But in the muscle, this may be a little different. So we may, instead of having different membrane potential, we may have different contractility, etc., etc. So here we have calcium release and then phosphorylation and activate of calcium binding proteins. But we also have the DAG activating protein kinase C. And protein kinase C is also activated by calcium increase. So these two pathways are complementary. Protein kinase C is similar to protein kinase A that we saw in the previous video about cyclic AMP, and it's quite simple to remember that because here we have phospholipase C and then protein kinase C. In the previous video, so cyclic AMP, we had cyclic adenylate cyclase, so A, cyclic AMP, and protein kinase A. So it was quite simple. Anyway, this is an overview of this pathway involving phospholipase C and diacylglycerol. I hope that this is helpful, and if it is, please give me a thumbs up and maybe consider subscribing to my channel. It will help a lot and it means a lot for me. Thank you. Until next time, I hope you have a great day and remember to study. Bye bye.